Okay, hopefully this is the last little bit. We've got, how many slides? About 40 slides. Okay, let's get it done. Sleep-wake cycles. Sleep is a state of partial unconsciousness from which a person can be aroused by stimulation. Cortical activity is depressed, but the brainstem is, pretty much remains the same. There's two types of sleep defined by EEG patterns. So there's the non-rapid eye movement, which is broken into 4 stages, and then there's REM, or rapid eye movement sleep. We pass through the first 2 stages of non-REM sleep during the first 30 to 45 minutes of sleep, and then we move into stage 3 and 4, referred to as slow wave sleep. Frequency of waves declines, but amplitude increases. EEG, heart rate, respiratory rate, blood pressure, and GI motility changes. At about 90 minutes in, 4th stage ends, and the REM sleep begins abruptly. Temporary paralysis, except rapid eye movements, oxygen consumption, heart rate, breathing increase can be greater than when you were awake. Most dreaming occurs in REM. The importance of sleep, very important. Slow wave sleep, or non-REM, stages 3 and 4, presumed to be restorative stage of sleep. People deprived from REM sleep get moody and super depressed. REM sleep may give the brain opportunity to analyze the day's events and work through emotional events or problems. This is when, it's like your brain does kind of off-gassing. It just kind of works things out. Also, eliminates unneeded synapses that were formed. So you dream to forget about traumas and to consolidate information. Daily sleep requirements decline with age. Stage 4 sleep declines steadily and may disappear at age 60. This is why elderly patients have trouble sleeping at night. Or they don't need to sleep, and so they're up super duper early and they don't sleep very much at all. Protection of the brain, you have the meninges. They cover and protect the brain and spinal cord. They protect blood vessels and enclosed venous sinuses. They contain cerebral spinal fluid. They form partitions in the skull. And they are 3 layers from superficial to deep: dura mater, arachnoid mater, and pia mater. Dura mater because the outside is very strong. Arachnoid mater because the middle looks like a spider web. And the pia mater because it's very small. Please know this diagram. It's this case, so dura mater is the strongest meninx, which is singular meninges. Meninx. Made up of 2 layers of fibrous connective tissue. There's the periosteal layer, attaches to the inner surface of the skull, found only in the brain, not in the spinal cord. Then there's the meningeal layer, which is the true external covering of the brain. It extends into the vertebral canal and as the spinal dura mater. The 2 layers are mostly fused, but separate in certain areas to form dural venous sinuses. The sinuses collect venous blood from the brain, empty into the jugular veins of the neck. The dura mater extends inward into several areas that form flat portions that divide the cranial cavity. Partitions are referred to as dural septa. That's plural for septum. They act to limit excessive movement in the brain. 3 main septa: the falx cerebri is it's inside the longitudinal fissure. Falx cerebelli is along the vermis of the cerebellum, and then the tentorium cerebelli is the horizontal dural fold over the cerebellum and in the transverse fissure. So when we look at the falx cerebri, got the tentorium cerebri and then the the falx cerebri. Couple things you should know there. When they say it's in your head, a medical term for something that is psychiatric or quote unquote in your head is going to be supratentorial, which means it's above the tentorium, which has to do with the brain. The arachnoid layer is the middle layer. It's spider web-like extension separated from the dura mater by the subdural space. This is where hematomas can occur because of the blood pooling there in that subdural space, a subdural hematoma. So subarachnoid space contains CSF, and it's the largest vessels of the brain are there. Arachnoid granulations protrude through the dura mater into superior sagittal sinus. It permits the reabsorption of CSF back into the venous blood. Pia mater is a delicate connective tissue that clings tightly to the brain following every convolution. Contains many tiny blood vessels that feed the brain. Which brings us to cerebral spinal fluid, CSF, which forms a liquid cushion of constant volume around the brain. It gives buoyancy to CNS structures. It reduces the weight in the brain by 97% by floating it, so it's not crushed under its own weight. Protects cerebral central nervous system from blows, impact, and other trauma. It nourishes the brain and carries chemical signals, and it's composed of watery solution formed from blood plasma, but with less protein and different ion concentration from plasma. As we mentioned before, the choroid plexus is a cluster of capillaries that hangs from the roof of each ventricle, enclosed by pia mater, and surrounding layers of ependymal cells. CSF is filtered from the plexus at a constant rate that equals that of reabsorption. Ependymal cells use ion pumps to control the composition of the CSF and helps cleanse the CSF by moving waste. Cilia of the ependymal cells help develop like a constant motion. Normal adult CSF volume is 150 mils, and it's replaced every 8 hours. Blood-brain barrier is the next thing we should talk about. It helps maintain a stable environment in the brain. Chemical variations could lead to uncontrollable neuron firings. Exceptionally impermeable tight junctions keep brain separated from many blood-borne substances and pathogens as well. Substances must pass through 3 layers before gaining entry into the neurons. Continuous endothelium of capillary walls. Thick basal lamina around the capillaries, and the feet of the astrocytes surrounding the neurons. Those 3 have to be bypassed in order for you to get to the neurons. Barrier is very selective, but it's not absolute. Allows certain nutrients to move by facilitated diffusion. Metabolic waste, proteins, toxins, most drugs, small non-essential amino acids, and potassium are denied access. Allows any fat-soluble substances to pass, including alcohol, nicotine, and anesthetics. Absent in certain areas, such as vomiting center and hypothalamus, it's necessary to monitor chemical composition and temperature of the blood. TBIs, or traumatic brain injuries, they include concussion, contusion, subdural or subarachnoid hemorrhages, and cerebral edema. CVAs, or cerebrovascular accidents, are also referred to as strokes. Ischemia. Now, ischemia is a tissue, or when tissue is deprived of oxygen or blood supply. It leads to death of whatever tissue, in this case, brain tissue, and it can be caused by blockages of the cerebral artery, by blood clot. Glutamate acts as an excitotoxin, which then, worsens the condition. Hemiplegia is, hemi is half, right? Paralysis on one side. Plegia meaning paralysis or sensory and speech deficits may result from a stroke. Transient ischemic attack, or TIA, is a temporary episode of reversible ischemia. So this is a mini stroke. Tissue plasminogen activator, or TPA, is the only approved treatment for a stroke. Clot buster. Big thing to note about the organization of gray matter in the spinal cord: it's reversed from the brain. Please, please, please know this diagram. So when you look at the spine, white matter, you've got is the myelinated tissue there. They run in 3 directions. There's one that goes up, one that goes down, and then one goes sideways, right? So there's the ascending to the higher centers. It's like sensory input, right? So if we want to send a message to the brain, you have to go up, right? From the spinal cord, always. Then you have descending from the brain to the cord, the lower cord levels, which is motor outputs, because we're going to do something about it. With what we're going to do something to compensate for whatever happened, right? And then the transverse is from one side to the others. These are commissural fibers. So white matter is divided into 3 columns on each side. So white columns or funiculi. There's the dorsal or posterior funiculi or columns, and then the lateral columns or funiculi, then the ventral or anterior white columns and funiculi. Each spinal tract is composed of axons with similar destinations and functions, and so please know that this piece here, you may need to know how to label it as well, like this one. The gray matter are actually called horns. So the ascending tracts and descending tracts, we'll go over this more in class, but it shows the actual location of the different tracts. I recommend knowing this picture, 12.29b. When we look at spinal cord trauma and disorders, spinal cord trauma, you can have transection. We talked about that. Paraplegia, if the transection is between T1 and L1, anywhere in the thoracic or to the lumbar vertebrae, you're going to have paraplegia, which is issues, with moving and feeling in the lower extremities only. Quadriplegia is transection in the cervical region. Then you lose mobility in all 4 limbs. Spinal shock is the transient period of functional loss caudal to the lesion. Ascending pathways, when we talk about ascending pathways, somatosensory signals travel along 3 main pathways on each side of the spinal cord. 2 pathways transmit somatosensory information to the sensory cortex via the thalamus. So when we talk about dorsal column, medial meniscal pathways, that's going to be the spinal thalamic pathways. These provide discriminatory touch, conscious proprioception, and how this is where we get sensory input. Third pathway is the spinocerebellar tract, terminate in the cerebellum. So when we look at the dorsal column, the medial meniscal pathways, lemniscal pathways, transmit input to the somatosensory cortex for discriminative touch and vibrations. It's composed of paired funiculus, cuneus and fasciculus gracilis in a spinal cord, and medial lemniscus in the brain. So, big thing, I need you to know the bold with this, the ascending pathways and the different pathways that exist. Spinothalamic pathways, lateral and ventral spinothalamic tracts, transmits pain, temperature, coarse touch, and pressure impulses within the lateral spinothalamic tract. So these types of questions are probably going to be more matching. Spinocerebellar tracts used to coordinate muscle activity. They have ventral and dorsal tracts, and they convey information about muscle or tendon stretch to the cerebellum. Here are the pathways. Please know that. For all of you all table people, there you have it. And just know that descending pathways and tracks, they deliver efferent, E for exit, impulses from the brain to the spinal cord. There's two groups, direct pathways and that go through direct pathways and indirect pathways. And know that the direct pathways are pyramidal tracks, indirect pathways are everything else. Motor pathways involve two neurons, the upper motor neuron, which these are the pyramidal cells in the primary motor cortex, and the lower motor neurons are the ventral horn motor neurons and the innervate skeletal muscles. The direct pyramidal pathways, regarding them, impulses from pyramidal neurons in the precentral gyri pass through the pyramidal, lateral, and ventral corticospinal tracts, descend directly without synapsing until the axon reaches where it needs to go in the spinal cord. In the spinal cord, the axon synapse with interneurons, which this is the lateral tract, or the ventral horn, which is the ventral tract. Direct pathway regulates fast and fine skilled movements. And here are the three descending pathways. Looking at indirect pathways, consists of four major pathways. We just need to know what they are: reticulospinal and vestibulospinal tracts. They maintain balance. Rubrospinal tracts control flexor muscles. Tectospinal tracts originate from the superior colliculi and mediate head movements in response to visual stimuli. Here are more pictures of the descending pathways this time, and tables for all of you table people. Okay, that brings us to the end of the lecture. I just want you to know what we're going to do in class is to kind of go back over and see what kind of questions you all may have. And the next bit will be to go over the cranial nerves and just going through the slides and saying, hey, this is important information, this is less important, or this is what's important from each of these slides. This should make our lecture go faster, and we'll be able to be more efficient about going through the lecture.